In the previous part of this lecture, we discussed about the innate immune response. Now, we will look at the adaptive immune response. So, as introduction to the adaptive immunity, let's first look at antigen receptors. So, we have several types of antigen receptors. So, one of them is the T-cell receptors. So, these T-cell receptors is used to discriminate between the self and the non-self. So basically, this is found in the surfaces of these T cells, and it helps these T cells identify whether that particular cell or particle should be attacked or not. And then we also have the antibodies. So it's another type of antigen receptor, but this is secreted by the B cells. Now, these antibodies, they do not directly kill the pathogens, but they can activate the complement system and initiate or facilitate in phagocytosis. So basically, they act as homing tags for the target, telling our immune system who to target. So actually, you have seen the antibody before when we talk about the complement system. So they help activate the cleavage of the C proteins to activate to initiate the cascade of reactions of the complement system. So here is a better view or a more close-up view of our antigen. This is the antibodies. So you have antibodies, basically a protein. It's not a cell. So this is a Y-shaped cell actually. So this is what it looks like in the ribbon model. So you have here, it's a protein. So you have here the coils and loops of the protein. So it can be viewed like this one. So it has a hinge region. So it's basically movable. So these arms are quite flexible. So you have a movable hinge here. So you have here the constant region and the variable region. Excuse me. This what is important in the antibody is this binding site for the antigen. This is the variable region. So this variable region is actually tailored to fit a specific antigen. So that's why they are called adaptive immune response, because they are used to adapt, or they are manufactured based on the actual virus or the actual pathogen. So for example, we have two types of pathogen here. So each antibody can bind to a specific type of pathogen. So they doesn't bind on just any pathogen. It must be specific to that pathogen. And that is also one of the reasons why this adaptive immune response takes days and weeks to actually activate, because they must first be able to identify. They need to study the protein makeup of the enemy pathogen and generate or develop an antibody that is specific to that pathogen. So we have here different classes of the antibodies. So we have secreted and membrane-bound antibodies. So when we name antibodies, we usually call them as immunoglobulins. There are more technical nomenclature for them. So IGs, immunoglobulins, and then we have different types, so A, B, C, D. So IgG and IgA, so these are the secreted antibodies. So they are released by the B cells. So IgG is the main antibody in the blood. IgA is abundant in the exocrine glands and secretions. So as you can see, they have their differences, just like IgA. We have here these are actually antibodies that are arranged end to end. And then we also have the membrane-bound antibodies. So IgEs are anchored to the surface of the mast cells, basophils, eosinophils, basically the innate immune responses. Because these innate immune responses, although they do not really they are part of the innate immune response, they also help in the activation, or they also assist in the adaptive immune response. And then we have the B cell receptors, so the IgD. So these are found on the surface of your B cells. So that's why these are called the membrane-bound antibodies. So they are not released as individual proteins in the blood cells or in the cells per se, but they are bound to the cells itself. Now we have IgM. IgM is also membrane-bound antibodies, but they can be secreted. They can be part of the secreted antibodies, but when they are secreted, they form this snowflake-like appearance. So IgM, why do antibodies have to have that polymeric appearance? You have the 5 Y antibodies that are joined together. So this also, because each of these parts, these arms of your variable region can bind to a pathogen, so it actually helps keep the pathogen in place. So it groups them and it minimizes the movement of the pathogens. That's also the reason why this IgA is arranged in such a manner, end to end, so it can hold at maximum of 4 antibodies. So it prevents the maximum of 4 antigens. So it prevents the antigens, the pathogenic, the antigen inducing or the antibody inducing pathogens, because the antigens are found on the pathogens. So I might use the term use the term pathogens or antigens in the same sentence. But anyway, they prevent the pathogens that they identified. They prevent them from basically spreading out. So they help keep the infection localized on a certain region. So that makes it easier for our immune system to actually mount a response, because when their enemy is on an area, on a localized area, it's much easier to fight them off versus their enemy running around the whole body where they have to chase them. Okay? It also minimizes the damage to the cells. So here are the antigen receptors, and then we have your actually this is more of this is a bit heavy on the genetic side, so I won't really delve too much on the production of your on this part of these antigen receptors. Now let's look at the adaptive immunity. So adaptive immunity includes two types of responses. We have the antibody-mediated immune response and the cell-mediated immune response. So both types of immune responses form effector cells. When we say effector cells, these are the cells that actually fight off the infections. So we have effector cells, that the lymphocytes that can act at once, and the memory cells that are formed to be held in reserve for the next encounter with the antigen. And that is why adaptive immunity is very important to us, because it remembers the type of bacteria or pathogen that enters our body, and so the next time we encounter them, it's easier to mount a response. And that's also why we need to vaccinate, because the vaccines initiate the production of these memory cells, so that we do not need to be exposed to this disease. We do not need to have that disease in order to have a proper adaptive immunity to them. So we already have the adaptive immunity without actually encountering the disease. So basically that's why we need to vaccinate. So here is a comparison of the antibody-mediated and the cell-mediated responses. So in terms of the effector cells, for the antibody-mediated response, we have the B cells as their primary effectors, whereas for the cell-mediated, these are the cytotoxic T cells and the natural killer cells, the NK cells. So for the mechanism, the antibody-mediated responses, they activated B cells secrete antibodies that recognize and bind to the antigen. So that's why it's called antibody-mediated, because its mechanism relies on the secretion of antibodies. Whereas the cell-mediated response, we have the lymphocytes, because that's why it's called cell-mediated. So it's basically the cells themselves that kill off, that gets the bulk of the work to kill off the infected and cancerous body cell. So what's the outcome? For the antibody-mediated, the antigenic molecules or particles are eliminated from blood and tissue fluids, because the antibodies that the B cells secrete, they initiate complement system, they attract the other cytotoxic T cells, they attract other white blood cells on the area of infection. So they actually, the antibodies more of, rather than killing off the pathogen, they serve as a signals that there is a problem here, attack here. This is also a homing, they also serve as a homing device, so that the directing the cells, the immune system, on who and what to attack. And then we have also for the cell-mediated, the outcome is of course the actual elimination of the pathogen along with the ailing cells. So for the cases, this is especially true in the case of viruses. Virus infect a whole cell, so they enter our own cells, infect them. So those infected cells are already compromised. So they must be eliminated by the cytotoxic T cells and even the natural killer cells. So the same way with the cancer cells. So they are bad. They are compromised. Are no longer good for our body. So they must be eliminated. So these are your the brief overview of the antibody and cell-mediated responses. So here is a look at the responses of our immunity, the adaptive immunity. So during the first exposure, so the reason why adaptive immunity is quite powerful, especially why it's important to have memory cells of that response, is because of this one. So here you have these naive lymphocytes who it was not yet exposed to any virus. So at first exposure here, this differentiates into memory cells. So part of them, a small portion becomes memory cell, and a larger portion becomes the effector cell. The effector cells that fights off the infection. So here is your primary immune response. So if you notice, it only produce a little amount of antibodies, and then upon the second exposure, you have the one that's are exposed here, of course, are the memory cells, and so they form more memory cells and much more effector cells. So this is the secondary immune response, and that is also one of the reason why we need to have actually most vaccines are come in two doses. So they have what you call the booster shots, which initiates or reinforces the memory cells, so that you can mount a greater immune response. So you will have a greater amount of the antibody against what the vaccine is for. For example, COVID vaccine of 2 doses. So the second shot actually gives you the much better boost. So just the first shot, you will get a small amount of antibodies circulating in the blood, it offers little actual protection to the virus. You need to have the second dose to actually get this full or a more effective protection against the virus. So you have this is during the secondary immune response after the second exposure, because the vaccines exposes us to these antigenic proteins of the virus, not the virus itself, but the proteins on the surface of the virus. These are the ones that elicits the antibody response. So basically, that's your adaptive immunity, and that's how we actually use them to our advantage, of course, in fighting off this pandemic. So defining characteristics of the adaptive immunity, we have the self versus non-self recognition by virtue of the T cell receptors and the antibodies. They're highly specific. So their responses are tailored to combat specific antigens. That is also why they act slowly, and that is why vaccines are the ones that are our tools to actually help our immune system protect us. So in terms of diversity, there are potentially billions of different antigen receptors. So they are much more diverse than the receptors found in the innate immunity. And one of the most important one is the memory. Our adaptive immunity remembers antigens through the making of memory cells. So how do they adapt, or how do they process the antigen? So basically, they need to be exposed to that pathogen. So they would engulf that pathogen, break them down, get parts of their usually protein coat, and then make them as present them outside. So present them to other leukocytes. So it's just like how in the barbaric times, you have your headhunters. So basically, they kill off their enemies and then use their heads as decorations, or parts of their armor as a decoration that, oh, I have killed this person, this is my enemy. So something like that. So they present it as an example to their comrades. This is our enemy. This is who we should fight. So here is a lymph node. The lymph node is somewhat like a headquarters or a base for our immune system. So specifically the leukocytes. So this is where the leukocytes are actually passes through during their patrols, as well as whenever there's an infection, they place the infection there, so that since it's a hub of the leukocytes, so that many other leukocytes will be exposed to that pathogen. When I say exposed, they would be able to identify who they should fight. So basically, when you have an infection or inflammation, you will see that the nearest lymph node to the site of infection is actually swollen. So that means that base, just like in warfare, that base or that headquarters is actually very busy doing the fighting. Okay, so that's it for this part of the adaptive immunity. We will continue on more on adaptive immunity in the next part of this lecture.